Okay, so when approaching our experiment, we discussed two different hypotheses. The first being our null hypothesis, which was that the independent variable has no effect on the dependent variable. And then to provide this in more specific terms for our experiment was that different colored LED lights do not produce varying effects on the growth of arugula. And then when we look at our alternative hypothesis, we discussed that the independent variable produces varying effects on the dependent variable. And then again, in more specific terms, different colored LED lights produce varying effects on the growth of arugula. And then diving into our background information, there was a lot that we wanted to discuss before we dove into our experiment. The first being that arugula, like most plants, appear green because they absorb all wavelengths of light except for green. And this is because plants reflect green light. Another thing to relate to this is that plants go through photosynthesis, which allows plants to utilize these wavelengths of light via their chloroplasts and produce sugars, which can aid and play a role in plant structures such as their cell walls, their leaves, etc. But mainly for our experiment, we really wanted to look at the idea of photomorphogenesis, which we discussed in lecture, deals with the growth processes that occur under light right after germination. And then in relation to this, the idea of phototropism, which relates to the directional growth of plants towards light when light is present. The main stimulus here with photomorphogenesis deals with blue light and auxin hormones, which is a signal that is distributed away from light. And something else to mention with photomorphogenesis is that it determines how a plant optimizes their light and space in relation to their growth, which we felt was very relevant to our experiment. And then the main photoreceptor for phototropism was phototropin. Another growth process, though, that we wanted to look at in relation to our experiment was the idea of etiolation, which deals with the growth processes that occur right after germination, but in the dark. And then the main photoreceptor here is phytochromes. And this is a photoreceptor that takes in red light and far red light. But red light is found in light conditions, while far red light is found in shaded areas or dark conditions. And something else to mention here is that red light is activates the phytochromes, while far red light inactivates them. So all of these we felt like were super relevant to our light experiment, especially after discussing them in lecture. And then pulling in similar experiments when looking to start ours was the first one we looked at was the growth of strawberries under different wavelengths of light. And specifically, they looked at white light, red light, yellow light, green light, purple light, and blue light. And what they found here at this experiment was that there was no distinction between the light colors when it comes to the fruit weight and overall fruit quality and growth of the strawberries. But something to note here is that strawberries do not go through photosynthesis. Another experiment was the growth of red cabbage under different colored lights. And here it was found that the highest germination and the biggest seedlings and overall dry weight was found under red and green light. And then a different experiment with tobacco also found that there was no clear color that favored growth for tobacco. And then the last one that we looked at was actually a student's experiment for a science fair. And they found that there was consistent and adamant growth under red light. But with this, we were really curious because there were so many conflicting results and ideas that really led us to want to conduct our own experiment, focusing on light growth, and specifically here, we chose arugula as our plant species. Thank you.